Hi, everyone. Welcome to the Ethel Browne Harvey Postdoctoral Seminar Series. My name is Leah Greenspan. I'm a postdoc in Brant Weinstein's lab at the NIH, and I will be moderating the session today with Ben Cox, a postdoc from UC Davis. We are excited to highlight the work of our outstanding postdoctoral members. Today, Dr. David Chen from NYU and Dr. Ceri Weber from UC San Diego will share their research. Each speaker will give a 20-minute talk followed by 10 minutes of Q&A. And if you can all please put your questions in the Zoom Q&A box after each talk. Our first speaker today is Dr. David Chen. Dr. David Chen received his bachelor's degree in life sciences and entomology at National Taiwan University. He then completed his master's degree in the laboratory of Dr. Chen Wang at National Taiwan University, studying the spatial temporal expression patterns of synaptotagmin isoforms in the developing rat retina. After receiving his master's degree, he pursued a PhD at UC Riverside under the mentorship of Dr. Anupama Dahanukar, where he researched the pharyngeal taste in Drosophila from periphery to brain. During his graduate training, he received the HHMI International Student Research Fellowship, as well as several awards, including the Dalil Nasser Award from the Genetic Society of America. Upon completion of his doctorate degree, he went to NYU for a postdoc in the laboratory of Claude Desplaines, where he investigates neural circuit assembly during brain development. He has received both an F32 fellowship and more recently a K99 R00 Pathway to Independence Award for his outstanding work. In addition to his science, he has also mentored many undergraduate students and taught several classes, winning an outstanding teaching award. It is my great pleasure to welcome David to the Ethel Browne Harvey Postdoc Seminar Series, and I am very excited to hear his work today titled Wiring Up the Brain During Development, Coordination and Propagation of Cell Fate Choice in Neural Circuit Assembly.
All right, thank you so much everyone for being here, and thanks SDB for this opportunity and Leah for this nice introduction. I'm a developmental neurobiologist. I'm really interested in understanding how the brain works, especially how you build a functional neural circuits. There are so many neurons in the brain, just like a computer chips. There are many different components need to be wired together precisely to ensure proper function. And not only there are many neurons, there are also many different cell types in the brain. As you can appreciate by this beautiful drawing of Cahal with the different morphology of those neuron cell types. So with the advanced genetics and imaging technologies, now we have an even better understanding of those different cell types, as well as their developmental origin, like how you pattern all this cell fate. So there are two main cell fate patterning mechanisms. One is deterministic. Everything is written in the DNA. It depends on what kind of genes or transcription factors a cell expresses. Then they're gonna become a specific cell type. A less known mechanism is called stochastic. It's just like rolling the dice. You don't know whether a given cell type will become A fate or B fate. It's kind of like stochastic. But this stochastic cell fate specification is actually quite common from bacteria to insect to human. For example, this is a Bacillus subtilis. The decision to enter a competent state is made stochastic. Only 20% of the bacteria will enter a competent state. As you can see, the GAP fused with the competence promoter. And also in the insect and human visual systems. So the decision for the photoreceptors or the cone cells in the human retina to express certain opsins or photopigments are also made stochastic. So with these two different cell specification modes, one is deterministic, another is stochastic. An important developmental biology question arises as how do those stochastic cells find their partners during development? So apparently fruit fly is a premium model because we know a lot about their color photoreceptor stochastic specification, which I'm gonna go into in a minute. And those photoreceptors, they are the periphery sensory neurons. They need to find their partners in the region in the fry brain called medulla. So over the past 20 years in Desplaines lab, we and many others, we actually contribute a lot about how those neural diversity are generated in this specific part of the visual system. So I'm going to summarize the past 20 years finding in just 2 slides. So what you are seeing here is a neural epithelium, which will later become a neuroblast, like a neural stem cell. As you can see the magenta colors here. And those are the stem cells. They're going to divide and produce a lot of neurons in green. And how you pattern different cell fates, it turns out there are 2 main mechanisms. One is called special transcription factors. It depends on whether which region expressed like red factors or green factors or blue factors, and they're gonna produce different types of neurons. And also when the neural stem cell age, when they keep producing the neurons and also renew themselves, they're gonna produce different temporal transcription factors through time. For example, first hour, maybe the red factors, and then green and blue and so on. So the intersection of these special and temporal transcription factors actually specify a lot of the cell type in this part of the brain, the medulla neurons. That's why I call them deterministic, because you can imagine it's like an address code, a zip code, where if you tell me a neuron is born on the dorsal side, first hour, it's gonna become a certain cell type. If it's the ventral side, second hour, it's gonna become another cell type. So it's very deterministic. So this is a nice model where we can ask how you find those two cell types to form the synaptic partners. So I propose there might be some molecular codes to help the neurons during development to find each other to pair the synaptic partners. How to find those codes? A few years ago in our lab, we actually used single cell RNA sequencing to generate all the gene expression profiles for all the optic lobe neurons. What you are seeing here is the adult fly brain. The regions highlighted in orange are the optic lobes processing all the visual information. So we dissect our optic lobes and go through the 10x single cell pipeline where we can sequence in every single cell and get all the transcriptomic expression profile. So this is the representation of those transcriptomes. Each dot in this plot is one cell. So we have all the gene information. And if two cells, they have a very similar transcriptome, they're gonna group together to form this so-called cluster. So one cluster can be presumably viewed as a one cell type. So as you can see from this map, there are around 200 different clusters. So that means there are 200 different cell types, and we have all the gene information for all these different cells. So presumably we can find the molecular codes through these data sets. So I'm gonna go a little bit detail about the stochastic part of the color photoreceptor specifications. So here is a fly eye. Fly has a compound eye. There are 800 different little eye units. So each dot is an eye unit, has 8 different photoreceptors. So we only need to care about R7 and R8 because those are the color photoreceptors, and those are the ones stochastically specified. So if you make a cross section, you will see R7 and R8, they actually sit on top of each other and detecting the same point of space. And they express different photopigments, rhodopsins to contrast the color information. So there are 2 different flavors of those eye units. One is yellow type. They're around 70% of the yellow type, around 30% of pale type. In the yellow type eye unit, R7 and R8 will express rhodopsin 4 and rhodopsin 6 to detect different wavelengths of light. And in the pale type, instead, they're gonna express RH3 and RH5. So that's why you can use those rhodopsin antibodies to kind of see this beautiful stochastic pattern of those color photoreceptor distribution. When I say stochastic, that means if you order number 1 to number 800 eye unit, if you look at the, let's say, number 200 eye unit across 4 different individuals, some of them will be yellow and others will be pale. That's stochastic. You don't know whether a given eye unit will be yellow or pale, but the overall pattern is like 70% to 30%. So those are the photoreceptors. The master regulator of this stochastic pattern is this spineless, as discovered almost 20 years ago from the lab. Spineless is transcription factors. What you are seeing in this circle is an eye unit. It's only expressed in yellow R7. So you can see all these spineless expression of eye units, they are yellow. The ones without expression will be pale. So in the wild type, you have a nice 70 and 30 ratio, but in the spineless mutant, everything becomes pale. And in the spineless overexpression, everything becomes yellow. So what you need to take away from this part is spineless is the master regulator to specify the yellow fate for both R7 and R8. Without spineless, the R7 and R8 will become the pale fate. So those are the photoreceptors on in the retina. So those are the sensory neuron. They need to find their target in the optic lobe so that when you make a horizontal section, that's where you can see this part, retina and the optic lobe, which are schematized on the right. So we have 4 different color photoreceptor, yellow, pale, R7, and R8. In the retina, they're gonna find their partners in the medulla, where we have a lot of knowledge about those neuron specification. So the main neurons I'm going to talk about is called DMA, the red neurons here, and also some other neurons called TM5A, B, C, TM20. Those are just the neural names because we know a lot about their morphology. We just give them a name, but these are just the medulla neuron, and they are specified in a very deterministic way. So how do those stochastic cells find the deterministic partners? So when we talk about synaptic partner matching, the predominant view is that during development, those two neurons, they need to project to the close proximity region, and then they're gonna find the right partners by the potentially use of these cell adhesion molecules on both sides of the membrane, like a presynaptic side, postsynaptic side. Those are the membrane molecules that interact with one another to facilitate the synaptogenesis, to form the functional synapse. So the key is actually finding those cell adhesion molecules. So one of the famous cell adhesion molecule families in the Drosophila is called DPR and DIP. So they are immunoglobulin domain superfamily. They use those IG domain to interact with one another, and there are 11 DIP, 21 DPRs, and they form very complicated interactons. But I just want you to pay attention to one pair, DPR11 and D gamma, because they are the interacting pair. Years ago, a previous graduate student in the lab, Max, discovered that when you look at the retina, you look at all these eye units, when he used the DPR11 reporter, he found that DPR11 is only in the yellow R7, because when we stay in the spinous is the yellow master regulator, we know this is yellow type. So when we look at the DMA cells, which are the postsynaptic cells, highlighted in red here, and the cell bodies on top of this cortex, so we blow up here, those red cells, they come in two flavors, one with the D gamma expression, and another without D gamma expression. Later, we call them yellow and pale DMAs, respectively. So the most important thing is, this is in adult. When we look at the development, remember we know the exactly address code for all these medulla neurons. We can use traffic gen or DAC, those two different transcription factors to locate where DMAs are during the larval stage, when the neurons are just produced. And on top of that, we use D gamma to see the D gamma expression. And I schematize the data here, where we found that there are two subtypes of DMA exist already. As you can see, this pale DMA and yellow DMA, even before the photoreceptor reach to the medulla. So that means it's not there's a naive DMA receiving yellow or 7 input and become yellow. And there are already two subtypes, while photoreceptor, they are figuring out their stochastic specification and try to project to the medulla. So how does that work? How do you connect the yellow R7 specific to the yellow DMA? It turns out the mechanism is quite elegant. What you need is just to produce way more DMAs. So you have, during development, you have a supernumerary DMAs, and then through the interaction of DPR11 D gamma in yellow R7, yellow DMA, the ones connected will survive, presumably provide some sort of traffic support. The ones without connection will just die. So that's how you can connect the stochastic color photoreceptor to those deterministic partners. But that's great, but we also have other three color photoreceptors, R8 and pale R7s. So how do they connect with one another, and how you build the whole circuits? So to answer this question, the first step is to understand what are the postsynaptic partners, those gray circles, what are the targets, what are the cell types of those targets, and then figure out the question marks, what kind of codes, what kind of cell molecules are used to pair those synaptic partners. So because of a time limit, I just tell you to address the first question, well, as a 5 researchers, we have a privilege to assess this electron microscopy whole brain connectome. So we can actually figure out the connectivity by looking at the EM connectome. And this is done by Mike Reiser and Matthias Warner's group, and it's published here. So basically we can trace all these postsynaptic partners and figure out their identity. Those are the ones I mentioned earlier, TM5A, B, C, and TM20 neurons. So it's much more complicated than that, because throughout the literature, we know there are a lot of interconnection between those cell types, but this is not the topic of this talk. So how about the question mark? What kind of cell adhesion molecules are used to pair those synaptic partners? I think to crack the code, it's easier to do it one pair at a time. And the most obvious candidate would be, well, how about the pale counterpart, pale R7 and pale DMA, because we figure out the yellow counterparts. So we have this transcriptome of a transcriptomic cluster of DMAs. So that means we have all the gene information of the DMA cells. And we can bioinformatically split them into two halves. One is D gamma positive, another is D gamma negative. So then we can ask the differential expressed genes to look for the ones enriched in the pale DMA, which is D gamma negative, And those genes should be important for pairing with pair R7. So, but looking for those molecules, you have to look at the right time because the synaptic partner matching happens during development, not in adult. So you have to look into the development. Fortunately, we not only have the adult transcriptome, we actually profile others developmental stages ranging from after pupillation, 15 hours, 30 hours, 40 hours, and so on and so forth, covering major developmental events from neurogenesis to epsilon pathfinding to synaptogenesis. So I decided to take a look at the P30 data set. And like I said, I split the DMA into two, yellow and pale DMAs, and look for the enriched gene in pale DMAs. And I only found two genes. One is called CG43737, and another is called B3C. So whenever you see a CG number, that means it's an uncharacterized gene in Drosophila. So these two genes not only enriched in pale DMA at P30, I check all the developmental time points, and they are all enriched in all these developmental time points. B3C caught my attention because it has been shown to be involved in exon guidance. So I decided to validate whether B3C is indeed expressed in the pale DMA. So what you are seeing here is the medulla DMA cell. It's located in the medulla layer 6, M6 layer. And I generate reagents to label B3C and use D gamma antibody to label yellow DMA. I hope you can appreciate that these two signals are mutually exclusive. That means pale DMA indeed express B3C. So what exactly B3C is? It belongs to another Ig domain superfamily cellulation molecules. There are 14 beats, and they interact with another type of molecules called site. There are 8 different sites in the genome. So as you can see, there is no non-binding partner from the non-interactome. So I decided to check all the site expression. Long story short, I found one site protein called site5 during development around P25 is expressed in subset of the R7 terminal, but not all the R7 terminals. But I'm not sure whether that's a yellow or pale. Until last year, we were in contact with the Grant Martin's group. They have this single cell data set for the retina. We only have the single cell data set for the optic lobe, but not the retina. And they have this during development at P40. I immediately look into this R7 clusters to look for the expression. So I first checked the spineless and DPR11, which I know they're gonna express in the yellow R7, and indeed site5 is expressed in the pale R7. And I can use a similar strategy to split the yellow and pale R7 to uncover more enriched gene in the pale R7s. So site5 is expressed in the pale R7. So far, I uncover all these molecules, and site5 is also unknown binding with unknown binding partners, so maybe they're interacting partners. So to validate those, the next step is actually doing the loss of function, gain of function of those candidate genes. But one important gap in the developmental biology is like we don't have a tool to label developing neurons so that we can do an RNA knockdown or overexpression. So part of my postdoc is developing this cell type specific developmental driver to label the developing neuron from early stage to adulthood. So for example, this is DMA cells. We have a DMA cells driver labeled early development all the way to mid-pupillation to adult, so that we can actually manipulate those cell types. So there are many outstanding questions. For example, whether the TM5A, also we know that this neuron expresses D gamma, whether this D gamma is also used like DMA to pair with the yellow R7, whether the same molecules are used for pre and post synaptic partner matching, and or this TM5C is received direct input from R7, R8, and indirect input from R7. If you mess up the connectivity on one pathway, will that affect other pathway? So with this color vision circuit, we can hopefully use electron microscopy, connectome, and transcriptome to find out the connectivity and the molecules, and also the tools I generate to really do the gain of function and loss of function study to validate those molecules, to kind of gain a general principle how you build a circuit. And hopefully this will be applicable to other systems like a human retina and mouse olfactory system where they are also specified stochastically. And with that, we can potentially know more about how circuits are built, and in the end, know a little bit more about how our brain works. With that, I want to thank my mentor, Claude, and also collaborator and past and current members from Developmental Labs, and most important, people who pay the bill, the funding agency, and also your attention. I would like to take any questions you might have. Thank you so much.
Well, thank you so much, David. That was a really great talk. Just to remind everyone to please put their questions in the Q&A box if you have questions for David. So I will get us started on questions. So I was curious, since you found all these really unique genes for the Y-DMA or the pale-DMA, have you yet started to knock down some of them? I know that's what you were saying, you're using your tools or overexpress some of them. And do you think that will lead to increased adhesion or wrong connections, or what do you predict you will see?
Yes, we are actually in the process of doing the loss of function. For example, I already generated a CRISPR mutant for Cy5 and B3C. The prediction will be if you lost all these acceleration molecule, maybe the pale DMA will miswire with the yellow R7. But there is also a possibility that the yellow R7 can still pair with the yellow DMA because they have right molecules. So maybe you have to do it in the double mutant backgrounds to really see the miswiring defect. So we are not only just doing the single gene knockdown, we're trying to do a knockout double mutants, for example, for both D gamma and B3C to see whether all these neurons are really confused to kind of wire their correct partners. But these are ongoing, yeah.
And when you do those knockdowns, is those global knockdowns, or are you doing neuron specific?
Yeah, we like to do that in a neuron specific way. That's why we generate a DMA driver. We actually not only have a pan DMA driver, we have a specific pale DMA driver along, and we also have a yellow DMA driver along throughout development. So we can perform the knockdown for those cell types specifically, not the global knockdown, because those molecules, they tend to express very broadly. So if you do a mutant, then a lot of cell types are also affected, and
Right, of
how to
course.
integrate our data. So we want to do it in a cleaner way. Yeah.
Great. Okay, so we have some questions from the audience. So Stephanie just kind of was surprised to hear that there were still some uncharacterized genes in Drosophila, since, you know, we feel like Drosophila has been around for many, many years. Are you planning on looking into that uncharacterized gene at all?
Well, that's probably not the first priority because you can see even with the many years of the work, we still try to figure out just candidates for just one pair. There are so many question marks here. So I think my priority would be trying to figure out all these question marks and then to see whether there are some general principle. For example, one hypothesis, maybe all the pale pathway they use beats and sides, and all the yellow pathway they use DPR and dip, because that's kind of like evidence from the current data, like why this beats and sides only in the pale pathway. Could it be all the pale pathway related neurons use beats and sides? Would that be having some application to other systems that could use two different families to specify two different functionally distinct neuron pairs? That would be something that I want to pursue first. Yeah.
Great. Ceri Weber says, great talk, David. Are the systems sensitive to changes in the other? I know it's a stochastic system, but does a change in photoreceptor ratio lead to a change in DNA M8 production?
Yes, indeed. There's a way to change the retina into all yellow or all pale. For example, the spine's overexpression will change every R7 into yellow R7. So in that case, then you will see more yellow DMA survived, and then the pale DMA numbers will reduce. So you do see the corresponding change in the DMA level. So we are trying to see whether we can see similar changes in the TM5A and B or TM5C level to see whether the cell numbers will change in response to the yellow and pale specification. But to do that, we have to label TM5A alone or TM5B alone or TM5C alone during development to really count the number of neurons, see how that changes. And we are in the process of getting all these very specific cell type specific driver.
What if you, just jumping on Ceri's question, what if you ablated certain ones? Do you expect, you know, not knocking down genes, but like getting rid of some of those cell types? Do you think that would affect the ratio then?
Yeah, that's a very interesting question. So like I said, when we try to ablate a DMA, for example, we have to have a genetic way to assess that neuron. So now we have this way of using DMA driver to drive some cell apoptotic genes, pre-apoptotic genes, to kind of ablate those cells. So, and then look at other cell types. But we haven't done that, but that's on the to-do list.
Great. We have another question from Anonymous. What is the deterministic versus stochastic? Why is the deterministic process changed to stochastic in photoreceptors?
Stochastic change. I'm not sure I fully understand the second part, but the way we describe it is the deterministic, meaning you have a very genetic programmed combination of transcription factor to specify those cell fates, and it's kind of robust across fly individuals. So whenever you see different fly individuals, the DMA is always coming from this part and specified by certain transcription factors. But those individuals, the precise color photoreceptors, whether it's gonna be yellow or pale, will be different across individual. That's why I call them stochastic.
And then I just have one more question, if that's okay. So it seems that although it's stochastic, you always have a yellow R7 with a yellow R8 and a pale R7 with a pale R8. Is there ever a situation where they would mix and match somehow, or that's just my naivety?
Yeah, so I didn't go into that detail because the spineless is only expressed stochastically expressed in R7. And then what's gonna happen is that when the R7 defines its yellow fate, it's gonna pattern the neighboring R8 cells through some other signaling. So I just abbreviate that. So it's actually R7 decide their fate and then tell the neighboring R8 to become the yellow fate. So there's no way you will get a yellow R7 pair with the pale R8 on the same unit. So it's initiated by the R7. Yep.
Great. Well, thank you so much for a wonderful talk. Hold on, I think we have one more question before we finish up. So someone asked, what does the arrow mean? Is there a link between transcription factor of photoreceptor to this transcription factor of medulla neurons?
Oh, those arrows just means the connectivity. It doesn't imply any sort of patterning or like transcription factor interactions between cell types. Those are just schematized of the connectivity, like who are the pre and who are the post-negative partners. Yeah.
Okay. Well, thank you so much for a great talk. And then Ben will take it over from here.
Yeah, thanks, Leah, and thanks, David, for an excellent talk. I actually started out in research and retina development, and so the topic will always have a special place in my heart. I'd like to introduce our next speaker, Dr. Ceri Weber. She graduated from the University of Washington, where she did a variety of undergraduate research, most notably in the lab of Billy Swala. There she studied evolutionary changes in ascidian development. She then moved to Duke University's program in developmental and stem cell biology, where we met, and I would say are the best place to study developmental biology, in my incredibly biased opinion. She joined the lab of Dr. Blanche Capel, and her PhD work there was on the topic of temperature-dependent sex determination in red-eared slider turtles. And this really fascinating research was published in Science in 2020. Since she graduated from Duke, she's been a postdoctoral scholar in the lab of Kim Cooper at UC San Diego. She also participated in the MBL embryology course at Woods Hole. I believe, if I remember correctly, was the first embryology course post-COVID. So I'm sure that was quite an adventure. So anyway, her work at UC San Diego has been on the establishment of vertebrate proportion and mammalian tail diversification, and that's been funded over the past several years by a Ruth Kirstein NRSA postdoc fellowship from the National Institutes of Health. I don't want to go too long on her current research, since that's what she's going to be sharing with us today. So without further ado, I will just let Ceri take over from here and tell us about mammal tails.
Great, thank you, Ben. And thank you, SDB, for hosting the seminar series. It's been really fun to hear the postdoc talk so far. So I'm here to tell you about tails and the mechanisms driving vertebral elongation and the evolution of axial proportion. So here I have a bunch of different mammal skeletons. Maybe some of them you'll recognize. And what I think is incredible about all of these is that there's traits that we see that they all have in common, like they all have skulls, they all have ribs, they all have vertebrae, and they all have limbs of some sort, which suggests that these features are really conserved. But there's also the fact that all of these bones look so different from each other means that they're also modular and evolvable. And what I think is amazing about this system is that all of these different forms have different functions that enable these animals to move and interact with their environments in incredibly different ways. From the cheetahs that are running and jerboas that are hopping, to hippos that fling their poop around and mark territory, or even communicating with others, like this dog wagging its tail. I have here one of my favorite examples of the diversity of skeletal proportion, where a giraffe, a dolphin, and a human, myself, and we all very obviously have different neck lengths. But this isn't actually due to the number of vertebrae in our necks. We all have 7 cervical vertebrae, but those vertebrae are all incredibly different sizes. So I'm interested in how the axial skeleton, specifically the vertebrae, are so diverse. How does that diversity develop, and how does it come to be through evolution? We know a lot about how the vertebrae are made during embryonic development. Genetic oscillations form these blocks of mesodermal tissue called somites, and the rate of oscillations determines how many somites are made, and those somites are going to eventually become vertebrae. So shorter bodied animals such as humans have fewer somites than, say, a snake that has a very, very long body and lots of somites. Those somites themselves actually resegment, so they combine together and then are cued to become different identities through collinear expression of Hox genes. So in a typical mammalian body plan, we have cervical vertebrae or neck vertebrae, thoracic vertebrae that are rib-bearing, lumbar vertebrae, sacral vertebrae that make up the pelvis, and then the caudal or tail vertebrae at the end of the body. And the best demonstration of the effect of these Hox genes is in this control animal here. Again, you see these thoracic vertebrae with ribs, lumbar, and then sacral with these little itty bitty bits of the pelvis on the side. And misexpression of Hox10 leads these lumbar vertebrae to take on a thoracic identity, whereas misexpression of Hox11 leads to the sacral vertebrae taking on a lumbar identity. So in sum, semitogenesis determines the vertebral number. Hox code determines the identity of those resegmented somites. And if that were the end of the story, maybe our vertebral skeletons would look like this, where all of the bones per region look exactly the same and are the same size. But that's not the case. In reality, even within regions, we have a huge diversity in the shapes and sizes of these vertebrae. So I'm interested in understanding how they acquire these different shapes and sizes, and how do individual vertebrae, especially neighboring ones, elongate and evolve independent of one another. I studied this in an amazing system that demonstrates extreme skeletal proportion. So the mouse, as everyone's familiar with in the lab, and the jerboa jaculus that everyone may know for having these incredible long hind limbs and feet. But they actually also have really interesting tails. Their tails are 1.5 times longer than mice normalized to body length, but they're actually composed of fewer vertebrae. So over here on the right, these are lengths of the vertebrae of the tail normalized to the length of their body. And here you can see that mice in blue have more vertebrae than jerboas do in pink. And most of this difference is actually made up by extreme elongation of these mid-proximal tail vertebrae that are clearly much, much longer than the tail vertebrae in that same region of the mouse. This pattern of short and long and short vertebrae, which you can see here also in these images of skeletons, has been described previously as a crescendo-decrescendo pattern, where you have these short vertebrae that get longer to a crescendo, and then they get smaller again to a decrescendo. When I started my postdoc, I was really excited about the jerboa tail and saw that they had this crescendo pattern. And I wondered how prolific this pattern was across mammals and rodents especially, and what other sorts of tail proportional patterns there might be. So here I have 3 example rodents that have very different ecologies: tree squirrels that live in trees, beavers that are semi-aquatic, and a Patagonian mare is a cursorial rodent, meaning it's just running around on land. And obviously they have very different skeletal forms that support these different locomotor ecologies. But if I look specifically at the tail, so I measure the lengths of the vertebral bodies in the cranial to caudal direction. And here I've represented those lengths as a proportion to relative to the longest. So the longest vertebra is in white and the shortest vertebra is in a dark gray. And the squirrels, as you saw before, have a crescendo decrescendo pattern where they go from short to long to short again. The beaver, though it has about the same number of vertebrae as the squirrel, have longer vertebrae for quite a while and only get short at the very end. And the Patagonian mara, with its much shorter tail, just goes from long to short. And indeed, we see these patterns all over rodents. Here's all of the squirrels, and they have this crescendo pattern that I described. Here are mouse-like rodents. So here is actually mus musculus in the rat right here. But their crescendos, you may notice, are actually a little bit more proximally placed than they are in the squirrels. And there's lots of examples of the short to long patterns scattered throughout the tree. And I've actually been measuring as many mammal tails as I can get my hands on across the country. The museums we've been to are listed down here. And we have over 250 tail skeletons now measured. And there are so many different tail proportions out there. We see this crescendo pattern in marsupials, even bats, even though they have teeny tiny little tails. Whereas the short to or long to short pattern I see in animals such as even in odd-toed ungulates like horses and rhinos and camels, or animals even like anteaters and sloths and shrews. So I think this incredible diversity in tail proportional length can actually be harnessed to understand how we get different vertebral shapes and sizes. So how does this develop, and then how is it diversified? We go back to the mouse and the jerboa, which are tractable systems in the lab. I collected mice and jerboas at birth, so that's P0 in this dark circles here in both of the animals, and collected them for micro CT scanning at every week until week 6 or P42 when they reach their adult size and proportions. So again, the number of the tail vertebra is on this x-axis, and the normalized vertebral length to the nasal anal distance is on the y-axis. At birth, both mouse and jerboa tails linearly regress from longest to shortest. You may notice that the jerboa doesn't have as many elements here. They just aren't as many ossified at this point in time, but both species have all of the cartilaginous elements that will make the vertebrae ultimately. And for both species, it's in this first week of life that we see this little bump, this disproportionate elongation of these mid-proximal tail vertebrae. And it's that pattern that is amplified through postnatal growth to make this crescendo decrescendo pattern in the mouse and an even more extreme crescendo decrescendo pattern in the jerboa. And I want to use this system both within the animals and between them to understand the cellular molecular mechanisms that drive vertebral elongation. I want to know how these different growth differences are controlled within a region of the skeleton. So these are all tail vertebrae, but how are these tail vertebrae growing so differently from one another? And how does that evolve? And then within the jerboa itself, I'm in a lab that primarily studies the limb. A lot of what we know about skeletal proportion has been studied in the limb. So how similar or different are the mechanisms that are driving these different growth patterns in the vertebrae to the ones that we already know exist in the limb? So vertebrae do elongate in the same way that long bones of our limbs do through endochondral ossification. So briefly, we have this mesenchymal condensation, and these cells will initiate differentiation to chondrocytes. Those chondrocytes will then start to rapidly proliferate and form these columnar cells that will then mature and enlarge through chondrocyte hypertrophy and secrete a collagen matrix that will become the scaffold for the mineralized skeleton. And I'll call this whole structure the growth cartilage in this presentation. So here is an example vertebra. This right here is trabecular bone. So this is the vertebral body. And then on the top is the cranial growth cartilage. So that's towards the head, and the caudal growth cartilage, which is going to be towards the tail or the butt end of the animal, if you will. And this all right here is the growth area. So if I were to zoom in on this, there would be different zones, a resting zone, proliferative zone, and hypertrophic zone. And this is maybe more obvious in a schematic. So these resting chondrocytes are ones that haven't yet initiated this rapid proliferation. The proliferative zone, PZ, is these stacks of proliferating chondrocytes. And then the HZ is the hypertrophic zone where they go through this enlargement. And the purple here is the trabecular bone. And it's the height of this hypertrophic zone that turns over every 24 hours. So the height of this is going to determine how much bone is going to be made per day, therefore the elongation rate. To make a longer bone and make a taller hypertrophic zone, you could simply just have more cells. If there's more hypertrophic chondrocytes, more of that zone is going to turn over per day. Alternatively, those cells could not change in number, but they just get much bigger. And that still makes a taller hypertrophic zone that can turn over. And then you could even combine the two of them together where there are more cells and those are bigger cells that create an even more dramatically taller hypertrophic zone. And indeed, in the limb, daily elongation rate is most driven by the size of these hypertrophic chondrocytes. So is that the case for vertebra as well? I wanted to compare vertebrae, therefore, that were similar to each other. They had similar rates of growth. So here I'm showing the relative change of vertebral lengths for every week from my micro CT scans. And in both species, mouse and jerboa tail vertebra 1 or TB1 are pretty similar. And then in this heat map, yellow is indicating the vertebra that is changing the most week to week. So that in the jerboa and the mouse is jerboa TB6. And it's not as apparent in the mouse, but they're on the same scale, which maybe demonstrates how incredible the jerboa's growth is. And then I wanted to compare these growths during the time in which they're changing the most. So mouse is changing the most in the first week of life, whereas jerboas are changing the most in the third week of life. I then used a calcium pulse experiment to measure the amount of bone that was made per day. And this corresponds with what we see week to week, where both the 6 tail vertebra in mouse and jerboas is growing more per day than 1, and jerboa 6 is growing more than the rest. To start to get at the broad mechanisms driving these differences in growth, is it more cells? Is it bigger cells? Is it more bigger cells? I can do simple measurements of cartilage in these histological images. So on the left, I have tail vertebra 1, and on the right, I have tail vertebra 6. On the left of each of those groups is mouse in the first week of life at both ends of the vertebra, and jerboa in that third week of life, P16 at both ends. And as we would expect, the height of the growth cartilage corresponds with the rate of elongation. I then can measure the height of the proliferative zone and find that this correlates with the growth cartilage height and rate of elongation. But interestingly enough, it's not due to the cells actually dividing faster in that zone, but rather there are just more cells present. So that supports the hypothesis that more cells make a taller growth cartilage. I measured the height of the hypertrophic zones and again found that this correlates with the height of the growth cartilage. But intriguingly, thus, the height of the actual hypertrophic cells in the direction of growth are not significantly different between the mouse at 6 or the mouse 1, or even that different from jerboa 1. So to summarize, jerboa and mouse tails at birth go from longest to shortest. In mice, this disproportionate elongation of these mid-proximal vertebrae is happening through simply having more cells in the growth cartilage. In the jerboa, this extreme elongation of these mid-proximal elements seems to be due to both the addition of more cells and bigger cells. So what are the genetic drivers of this difference? I've used the same comparison that I showed you before for an intersectional RNA sequencing experiment where I compared jerboa 6 to mouse 6 at these most rapid changes of growth. And then I compared jerboa 1 to mouse 1 as a reference for tissues that are doing the same thing at the same time. So we did the differential sequencing or differential expression analysis between 6 and 6 and 1 and 1. And on this chart here, I'm showing you all genes that are differentially expressed, significantly differentially expressed in both groups. So on the y-axis is tail vertebra 1 to 1. Positive values indicate a gene that is expressed higher in the jerboa relative to the mouse. And on the y-axis, we have jerboa 6 to mouse 6. And again, a positive value indicates a gene that's expressed higher in the jerboa relative to the mouse. And here we plotted this line, and all of the dots are different genes, and dots that are gray and along this line are equivalently differentially expressed in both. And that won't explain a disproportionate growth difference. So I imposed a 95% prediction interval and selected the 421 genes that lay outside of this line as ones that are disproportionately differentially expressed and might explain these growth differences. An even more extreme version of this comparison would be to only look at the differential expression analysis of the 6 to 6 comparison. So these are genes that are significantly differentially expressed between these two in different species, but not significantly different when 1 to 1. So on the x-axis again, we have jerboa TB6 to mouse 6, and positive values indicate a gene that is expressed higher in the jerboa 6 compared to 1. And that analysis identified about just over 1,800 genes. So now that I have a reference and I have genes that might be driving the disproportionate elongation of this TV6 between species, I wanted to then look at the genes that are differentially expressed within species between 6 and 1. And I chose the most extreme, the Jerboa 6 to Jerboa 1. And that's what all the genes that are plotted on this here. I then have actually color matched this. So I intersected this interspecies analysis with my interspecies analyses and have highlighted genes that have a coordinated sign change in log twofold expression, meaning that if the gene is expressed higher in the comparison of Jerboa 6 to Jerboa 1, and it's expressed higher in Jerboa 6 compared to mouse 6, it's going to be orange. And if it's lower in both of those data sets, it's going to be colored purple. And we identified 1,400 candidate genes in that group. And some of them you may recognize as being critical to bone development. Now, back to my previous question, we know a lot about skeletal elongation and proportion in the limb bones. How do the vertebrae genes compare to what we know about the limb? So you can imagine all of these 1,400 genes that I've identified is in this pink circle. I first intersected it with an analysis that we previously did in the limb that was in a similar intersectional RNA sequencing analysis and found that there is significant overlap between the two, which suggests that Jerboa skeletons are utilizing the same genes in the axial and appendicular skeletons to get this extreme elongation. I also intersected it with a data set that was looking at differential senescence. So these are going to be genes that are differentially expressed in a growth cartilage that is nearing the end of its growth, so it's growing slower versus when it's most rapidly growing, or genes that are between growth cartilages that are at the same point in their lives, you might say. They're rapidly growing, but they're growing at different rates. And again, there was a significant overlap. Finally, I was interested in proportion, especially. So here, this is a genome-wide association study looking at the height of a sitting height of a human compared to the lengths of their limbs for the human sitting height ratio, and found significant overlap. But what's really incredible is if I overlap all of these together with my data, there's actually only one gene that shows up consistently in every single data set, and that's natriuretic peptide receptor 3. And the natriuretic peptide signaling pathway is known to be critical to bone development and skeletal growth. NPR2 is a receptor that binds the circulating C-type natriuretic peptides. It leads to synthesis of cyclic GMP and activation of genes that are going to drive chondrocyte proliferation and differentiation. And indeed, loss of this gene leads to skeletal dwarfing, and it's intersecting and interrupting FGFR3 signaling. In contrast, FGFR3 is considered to be a clearance-mediated receptor, so it binds these CMP peptides and leads to degradation, though there is some work that suggests there may be an independent function. So we wanted to know, how does the loss of NPR3 affect vertebral proportion specifically? NPR3 knockouts have been studied before, and actually before they even knew it was NPR3, it was this mouse that had elongated skeleton all over its body globally, and they were called long john mice. And you can see that the limbs and the vertebrae, they have the skeletal kyphosis, all get much, much longer, but they hadn't investigated the proportion of the vertebral skeleton. So we obtained these mice. So here at P42 is a wild type mouse on the left, and then here is its mutant sibling with its characteristic kyphosis, and it does have a longer tail relative to the length of its body. So here we have NPR3 mutants are these orange triangles, and NPR3 wild type siblings are these gold blocks. And this blue line here are just the CD1 micro CT scanned mice that I showed you earlier in the presentation. What's most interesting, though, is we did a micro CT scan of these skeletons too. So again, we have the wild type sibling in gold and we have the mutant in orange. And it seems that the proximal tail skeleton is disproportionately taller than in the wild type. This would suggest that NPR3 does have a role in driving the proportion of this crescendo decrescendo, and maybe it's only really affecting the proximal tail skeleton as opposed to the distal that looks similar between the two. And from that previous study of the long john mice, they identified that these differences in the limb skeleton were at least from this taller hypertrophic zone. So perhaps this is happening through a change in chondrocyte hypertrophy. Now, many of you may say, whoa, Ceri, your boss just wrote a paper about how simplistic explanations of evolution may not be honest to the data that I've collected. And that's definitely true. I don't think the end of the story is MPR3, but rather we think that it's one of many genes that are modularly contributing to changes in skeletal proportion. So I have my data here of our candidate genes. And I already said before, there's some obvious regulators of skeletal development already in here. For example, SOX9, FGFR3, and NKX32 are really critical regulators of chondrocyte identity and maintaining a chondrocyte pool. So perhaps regulation of these genes is important in the number of cells in the growth cartilage. Alternatively, we see Wnt4, Indian hedgehog, and MPR3, which are known to have roles in chondrocyte differentiation and hypertrophy. So perhaps regulation of these genes are modulating the size of the cells. And finally, there's a Hox gene in here. And so maybe this is important in cueing where in the tail this disproportionate elongation is going to be happening. And those are just the genes that we already recognize and have characterized. There's many, many more in here that may be contributing in different ways to different skeletal proportions, which brings me back to our museum data sets. In a collaboration with Talia Moore and Chris Organ, we're working on defining the locomotor and tail usages for all of these different animals across the mammal tree so that we can do phylogenetic analyses and determine evolutionary drivers of tail diversification. And I'm really, really excited to dig into all of that data. And I say that we're cataloging the tail use types because we do think that these different proportional patterns are functional. So jerboas, when they move in the air, they use this thing called inertial maneuvering to position their body in space, and their tails are helping them accomplish this. So in collaboration with Talia Moore's lab, we've made a simulation where the body here is this rectangle and the tail is this rod. And that simulation is having that move in space through inertial maneuvering. So the first question was, what are the optimal number of tail elements to inertially maneuver in space? And it turns out more elements helps a tail move the body better. The next question was, how about the size of those elements themselves? If a different combination of short, long, short, or any other one optimizes inertial maneuvering? And it turns out, indeed, there is. The optimal configuration when you have 4 vertebrae is a short, long, short, short pattern. And if we look in the museum data sets, we find that animals that already are described to have inertial maneuvering have a much taller crescendo amplitude than generalist tails that have a much smaller amplitude. So perhaps the height of this crescendo is optimizing their inertial maneuvering. So with that, I'm really excited to continue digging into the molecular mechanisms that are driving axial skeletal diversity, understanding how that evolves and how those different processes can drive evolution of tail functions. And I'd like to thank my lab, particularly the people in bold for helping out with my preprint team, tail, micro CT scanning, my mentors, IGM for the sequencing, and then the funding sources that supported all of this work. And with that, I will take any questions.
Thanks, Ceri. Really awesome talk. We can get into questions here. First one from Stephanie Granger. Do you see any regulation of CDX genes, which are upstream of Hox, in your comparison?
I'm trying to think. I don't think so, but I also haven't specifically searched. I will probably do that right after I finish talking here. There's a few other Hox genes that show up in different intersections that we've done. But I can't remember if I've seen a CDX, but that would be really, really cool to check.
Cool. So got a lot of questions actually, so I'm going to go through them as quickly as I can. From David, great talk, Ceri. Your intra and interspecies RNA-seq comparison kind of assumes that the factors influencing vertebral elongation are cell intrinsic. Could it be possible that the genetic drivers of disproportionate vertebral elongation are instead secreted or gradient factors, which might not be detected by your current approach?
Yeah, I mean, obviously the MPR system is driven by a peptide that is circulating and not necessarily synthesized in the growth cartilage. That definitely could be the case. I'm actually planning on doing a single cell sequencing experiment that might get at more of this. But I do think that those peptides and those signaling factors have to bind to something. And so looking at these differences in expression that are already apparent maybe have larger effects, and these circulating peptides or growth factors or things that are being expressed up and down the growth cartilage may have effects in other pathways that we see downstream changes in. But that would be really cool to find out.
All right, keeping going. Just from Guy, naive question: what limits the size of the tail? Why don't they just grow forever?
That's a great question. Obviously, towards the end of the tail, there actually is a little bit of variability in the number of vertebrae. It's like plus or minus one. And that's thought to just be at the end of the tail, you're just burning up mesodermal progenitors, and the tail has to stop somewhere. And so I don't know if it's just, I mean, obviously some species make 40 tail vertebrae and some make 10. Every single animal makes at least some. So even people like humans or great apes that have internal tails, we still make like 4 tail vertebrae. And so I would assume at some point, this would be a cool hypothesis to test with our biomechanics collaborations. Is there an optimum tail length? And there doesn't need to be any longer biomechanically to achieve what you need to achieve. Does it become like a hindrance at that point?
Okay, last two. In the from Will, in the NPR3 knockouts, what was the growth of other tissues, or sorry, was the growth of other tissues in the mouse affected in a similar way? For example, skin on the tail, was it tighter on the knockout mouse?
Yeah, the NPR3 knockouts do have disproportionately elongated limbs, and there does seem to be a proximal distal effect that's been described before, although I haven't measured it in my mice. The tail definitely feels different. The skin is a little bit tougher. I don't know if it's because it's tighter. I haven't looked at that, but like when I'm collecting these animals and degloving the tail, it's harder to cut through them. It's also really interesting. This is maybe a little graphic for people who haven't worked in mice, but when you are euthanizing a mouse and you grab the base of the tail, in the NPR3 mutants, you can actually feel the knobs of the transverse processes. And in a normal mouse, you can't feel that. And I don't know if that's because the entire skeleton is shifted down into the tail. And that might be the case actually in where is it? Here, you see like a little bit of the fur. They call this the tail cone descends deeper into the tail. And this may be because all of the skeleton is longer and it's pushing it down. Maybe the vertebrae themselves are growing out. Haven't described that yet, but I think that'd be really cool to check.
All right, last one. From Karina, great talk. Carrie, two-part question. Do you think these factors are involved in regulating the rate of cells exiting or sustaining being in the cell cycle that contribute for the number of differences you see in your research? And then 2, have you or your lab started to explore the factors you've identified in your research in the context of regeneration?
So for the first part, like the factors like NKX32 and SOX9 was a kind of a cool finding, because I've been wondering if the ability to sustain growth for much longer may just be because they have a pool of cells to keep pulling from to initiate proliferation. I also, everything I've done right so far has been a point in time or like 2 days in time. And I don't know any of the growth parameters. Like, is there an initial difference in proliferation as the growth plate initially becomes very large? Because it doesn't start out much bigger than the mouse. They are quite similar and then become very different. And I think that would be a really cool experiment to do. It'd just be like an EDU pulse experiment and collect animals up until these growth points to see if there's an initial burst in proliferation and how these cell numbers change. And then for the second part, exploring the factors, I haven't yet explored, besides NPR3, everything else is still an open question. And I haven't looked at these in the context of regeneration, though some of these HOX genes are involved in regeneration. A lot of these genes come up in like the axolotl and other sorts of tail regeneration pathways. Those would be cool intersections to do actually and see.
Great. I'd love to talk to you about like ECM and stuff in here, because that's my interest, but we're about out of
Yes.
time. So
We'll talk.
I can talk to you offline. But anyway, so thanks to David and Carrie for excellent talks. This seminar has been recorded and it will be available on the SDB website next week. So please come back and join us for next month's seminar. It will be Friday, December 13th. And Ashley Libby from the Francis Crick Institute and Silas Boy-Nissen from Stanford will be presenting. So thanks to everybody for coming out, and have a great rest of your weekend.